Friends, today's NRI Samay program from Los Angeles brought to you commercial free by listeners like you. Your generous donations make possible for us to stay commercial free, and today's show is pre-recorded. If you have a question for the guest, please send an email to nrisamay@gmail.com. We will follow up with the guest and find the answer for you. Thank you for your support.
Friends, this is your host for today's show, Srihari. Today we have an incredible person. He's called a seed hero. His name is Dr. Debal. He is a scientist, ecologist, and he has worked to preserve about 920 strains of indigenous Eastern Indian rice, which are now freely available for everyone to use and stored in seed banks in West Bengal and Odisha. We'll be talking about the importance of keeping the traditional seed and the dangers of the genetically engineered seeds. Dr. Debal, welcome to NRI Samay. Dr. Debal,
yes.
Could you please tell about the GM seeds, whether it is necessary for us or not?
Yeah, my take is that genetic engineering and the products of transgenic production of crops, novel crops, with certain characteristics which might be beneficial to the farmers or consumers, is absolutely unnecessary from agricultural science point of view. The reason being, there are two types of arguments. First is that the characters for which these transgenic crops are being produced or being researched into are already present in most of the crops that we have been using all over the world. For example, if we consider rice, with which I am most intimately, most deeply concerned and involved with over the past 17 years of my research, shows that we have certain characteristics of rice, especially in the indigenous traditional land races of rice, of thousands of varieties, which are not yet being incorporated in any modern varieties successfully. So one example is deep water tolerant rice. Another is submergence tolerant rice. Another is salt tolerant or salinity tolerant varieties of rice. Now all these properties are already present in rice varieties. So the genetically engineered rice which can tolerate high amount of salinity in the soil is therefore unnecessary, because that gene and that property is already present in many of the varieties which are being or were cultivated by thousands of farmers in the past in West Bengal, for example, in the Sundarban ecosystem, where there were regular episodes of saltwater incursion. Now, despite 60 years of rice genetic research, rice geneticists have not yet been able to develop any modern variety which can successfully tolerate high amount of salinity in the soil, or can survive in a 10 feet deep water, in a land which is being inundated by 10 feet deep water and holds that water for 3 months. So this salinity resistance or salinity tolerance or flood tolerance of those rice varieties, submergence tolerance, some of the genes have also been identified. We know that the rice genome has been mapped, and we have also identified several genes which are responsible for or which can impart these properties on the rice plant, like SMB gene. SMB stands for the submergence tolerance. That also has been incorporated into some modern varieties, but when it's put into the farmer's farm, that doesn't succeed. In West Bengal, there has been one submergence tolerance plant that was introduced about 40 years ago, but this has failed to successfully produce any of these. They can't even survive in deeply submerged areas. So that shows that we need to conduct even deeper research, more intensive research in the functional genomics, not just the structural genomics, but functional genomics, which type of genes and gene combinations in combination with the environmental conditions would be successful in which type of land and climatic conditions. So instead of investing in that kind of research, a functional genomics of the genes which are already available in the rice plant, is completely unreasonable, and I would say unintelligent to invest into a research which requires 100 times more investment for identifying a similar gene in another type of organism or even another kingdom like bacterial kingdom or animal kingdom, and then incorporate that gene into the rice plant. So that's why it is not necessary, because we already have some of those properties for which transgenic genetic engineers are searching for. The second argument is that there are certain properties which may or may not be available in a rice plant. For example, the golden rice. The golden rice is propounded as a rice which is fortified with vitamin A. Now, nobody claims that rice is the ideal food, like milk, that every vitamin will be available in the rice plant or rice food. But the rationale for incorporating vitamin A into rice is that the poor people in the South, in global South, will be benefiting from rice, which is the staple. And if it incorporates a substantial amount of vitamin A, then that would be a good supplement, dietary supplement to as a solution to the vitamin A deficiency, which is a major health concern. Now, if we are actually really targeting the poor population who have no access to a substantial dietary supplement of vitamin A, then just eating rice is not going to solve the issue, because it's much more complex than this simple supply of nutrients. First of all, even if there is substantial amount of vitamin A available in the diets of rice, it may not be absorbed or assimilated in the body of the poor people in the South if there is an endemic diarrhea or dysentery in that area. Because whenever a person has a diarrhea, then no amount of vitamin A or vitamin E can be absorbed in the body. It will be immediately flushed off the intestine. So we have to take care of this, the water supply and the safe water and the other health security and infection, coliform bacterial infection and dysentery, anemic dysentery infection, and so on and so forth, without which no amount of vitamin A, whether from milk or whether from rice or any other sources, are available. This is, for the major social health, social hygiene and health question. But the other point is that for the access of cheap availability, easier availability and access of vitamin A, our poor people in the South have already got this access, because we have hundreds of uncultivated wild plants and animals, and also some of the cultivated crops, which already contain enormous amount of vitamin A, and which could be 200 to 600 times higher content in vitamin A than the best quality of golden rice, which is the third generation that's going on. In the third generation of golden rice, which is now being developed in India also, contains not more than 200 μg, 200 microgram per 100 gram of rice. And if you compare, for example, Ipomia, there is no English equivalent of that plant, Ipomia aquatica, which is an wild edible plant, which is freely collected and harvested by the rural population in India, contains more than 1,100 microgram per 100 gram. Similarly, if we consider another uncultivated plant, uncultivated yams, like diascorid yams, some of the diascorid yams and Amorphophallus and Colocasia, these are the plants, all of the yam group of food. The shoots, young shoots of Colocasia, for example, and some other yams, also contain something like, vitamin A ranging from 800 micrograms to 1,300 micrograms per 100 gram of the food, which is again more than 600 times higher in vitamin A content than the golden rice. And in each case, I'm giving examples of the plants, the food plants, which are uncultivated, which means it requires no investment by the consumers. And anyone can, get it for free from the forest or from the waterfronts, wetlands, or even their, farm margins, free of cost. Now, instead of making these foods, wild uncultivated foods, and some of the cultivated foods like pumpkin is also a very rich source of vitamin A. Instead of making these more accessible and more easily available to the poor population, investing billions of dollars in developing an esoteric genetically engineered food, which is may not be accessible to the poor at all, is really misplaced investment and is not really intelligent food policy, either from science point of view or from political point of view. That's it. So these are the two reasons why I think these genetically modified seeds of any food is completely unnecessary, because we already have in stock both the number of types of crops and uncultivated foods available. And within the cultivated flora, we already have a range of characters for which genetic engineers are searching.
Friends, NRI Samay is a non-profit independent alternate media from Los Angeles, USA, which brings positive stories from grassroots activists from all corners of India and across the world. Among others, we focus on stories of need for equality, inspiration, environment, health, and politics. NRI Samay does not rely on corporate funding, and thus our shows are commercial free. We only rely on your donations. Please consider donating to NRI Samay, and all donations are kept transparent on nrisamay.com.
Sir, just to expand on that, one of the arguments is that the research should continue because of the environmental changes and also the explosion in the population, and we don't know how it will look like in 50 years or 100 years. So we need this research is what one of the arguments is. Could you comment on that?
No, it's a different argument. I mean, nobody says that genetically modified food should not be accepted. Does not mean that research in genetic engineering or genetics should be stopped. This is not saying that. I mean, this is like, arguing that in order for understanding atom, we have to manufacture and use atom bombs all the time. That kind of argument. So this is not the argument to support genetic engineering in agriculture. We have already benefited a lot from genetic engineering. For example, genetic engineering as a tool to science. For example, we are now able to manufacture transgenic insulin, which has become very cheap, much cheaper than it was about 50 years ago. But that's why we are definitely not against genetic engineering per se as a tool of science, which could be predictably and manageably used in the human economic systems. So, in the case of insulin, if we take this as a paradigmatic case, if there were any evidence that the transgenically produced insulin has any side effect or unpredictable effect, which were not very conducive to human health, then we have the means to stop the production of insulin immediately in the lab, because we have this means and we have absolute control over the manufacturing, because it's the micro location where these are produced. But similar thing cannot be told of GM crops, because once that crop is released in nature, there is no scientist or no power in the world which can call back all the pollen, for example, or all the seeds from nature. So we cannot have any kind of means of calling them back and push all the pollen from GM crop that was introduced in nature back into the laboratory. So that's why we have to be extremely cautious before releasing them, and we have to be very cautious about these, all types of long-term effects, whether it's human health or the environment or biodiversity and everything, before pushing it. And that's what this precautionary principle says. Precautionary principle does not say that we should stop research. Precautionary principle, whether it's in medicine or chemistry or ecology, it only says that we have to take extreme caution with the statistical type 2 error being the guideline. There are two types of errors. Type 1 error, which shows that the effect is not there, but we hypothesize that there is an effect. And type 2 error is just the reverse, that there is an effect, but we presume that there is no effect. In that case, if there is really a harmful effect, a toxic effect of any food product, for example, and we presume that there is no effect, this is type 2 effect. It's a type 2 error in statistics. Now, if we make this kind of effect, and after 50 years from now, people discover that there was this kind of, severely harmful effect from that, then we may have sacrificed, hundreds or thousands of lives already. And therefore, the precautionary principle in medicine or chemistry always says that we have to reduce this type 2 error. That's the target. That is precautionary principle. And very unfortunately, this precautionary principle is followed in chemistry and medicine. But when it comes to transgenics or genetic engineering, nobody talks about that, and everyone says that we are going to lose the benefit. This is really ironical, simply because today's transgenic research is completely controlled by the corporations, who are motivated not by science, nor by humanitarian ethics, but only by profit maximization ethics, nothing else. And that's why they want to maximize the profit in a minimum possible time without giving recourse to any kind of scientific precaution and scientific philosophy of type 2 error reduction.
I see.
So, therefore, it is completely, unconnected to the question of stopping research.
Friends, NRI Samay is a non-profit independent alternate media from Los Angeles, USA, which brings positive stories from grassroots activists from all corners of India and across the world. If you have any interesting episode you would like NRI Samay to cover, we will be happy to do so. NRI Samay would also like to encourage amateur journalists who have a flair for highlighting current events. You can be part of our citizen journalist team. You could cover an important event, interview victims of an atrocity, or document an inspirational story. The idea is to have a mix of experience throughout all parts of the world, preferably supported by an audio or a video clip of your story to be aired, which will be the curtain raiser. You can also submit your opinion articles on important topics to NRI Samay. We will be happy to put it on nrisamay.com in the opinion section. All your work will be credited only to you. And if you're interested, please send an email to nrisamay@gmail.com.
I see. And is there any evidence that this transgenic methods or seeds are bad for human health or environment? If so, what are they, sir?
There are three types of evidence that came up in the scientific literature. One is the general effect on the agroecosystem, or you can say ecological effect, on biodiversity, for example. And biodiversity, that includes species diversity, species of different other crops and other animals in the agroecosystem. Second is effect on the human health, and that is toxic effect or whatever. And the third is the effect within the plant itself, which is being controlled, which is at the cellular and molecular level. There has been dozens of papers which have already indicated the different kinds of unpredictable effects on the plants themselves, on the transgenic plants themselves. For example, there are gene silencing or overproduction of the gene products or underproduction of the gene products or differential production of the gene product, transgene product, in different tissues of the plant, which are all unpredictable. Even gene silencing has been observed in many transgenic crops, including BT cotton and BT plants, where the BT, that is Bacillus thuringiensis bacterial protein, and that nomenclature is CRY1A and CRY1B and all these types of proteins. These proteins are completely not produced, and that's called gene silencing. That has resulted in the crop failure of crop protection. The other type is that differential production of protein in different tissues. So in the BT cotton, for example, the gene may be successfully produced in the leaf or in the root, but not in the cotton boll, resulting in that it's completely destroyed by the cotton pests because the boll is all eaten up. But because it develops in the cotton root, it has no effect on the pests. So that is one that has severe agronomic consequences. The second part, that is effect on biodiversity, are of various kinds. One is that the toxic pollen, if it's physically transferred to other, simply by aerial displacement, it lodges on some other plants, which is the food of other organisms like larvae of insects. They might be killed off. There are certain studies which have also indicated that lacewings have become adversely affected. Lacewing population has declined because of this kind of toxic effect of BT corn. And some lepidopteran, that is butterfly or moth species, which have also become, the population has reduced because of this toxic effect. There is also an evidence of horizontal gene transfer into other organisms like rice to wild grasses or wild ancestors of grasses, wild population relatives of rice. They have been, the transgene has been transferred to those other organisms, which is also unintended and unprecedented. That evidence also has been documented several times over. There are also recent studies which indicated that transgenic corn pollen has also transferred this transgene into traditional varieties of corn in Mexico, even across 500 kilometers in distance, which cannot be controlled by any kind of technology that way. And so there are so many evidence. There is also another study that was published in Proceedings of National Academy of Science, USA, that showed that the BT toxin washed away from the field, BT corn field, also has adverse effect on the aquatic organisms. And then there is also an effect of this. That was a study of long-term study in Vrihi that was published in 2005 in the Proceedings of Royal Society of Vrihi, and that showed that article was the lead author was Bohan, and Bohan's study indicated that after 5 years of study, they proved that the cultivation of transgenic herbicide-tolerant crops, mainly soybean, that has eradicated all the lepidopteran population and broadleaves because of the excess use of herbicides. And because of this, use of herbicides management in the transgenic crops, so this is an indirect effect of this herbicide thing. This has eliminated this biodiversity too. Both the broadleaved plant population and the lepidopteran population, which depend on these broad-leaved plants for their production and for their reproduction and breeding. So these are the evidence. On the health effect, there is just a few studies, including the recent ones by Professor Srihari. And just last week, his Srihari study was suddenly revoked by the editor of the journal where he had published for completely flimsy grounds. And coming to that, with the lengthy things. The problem is that, the effect, human health effect or general health effect on animal models of the crops, transgenic crops, is so few simply because the companies have the proprietary authority on the crops that they generate and market, and they make stringent conditions on the scientists, whoever wants to study. That includes me. When I wanted to study this, the ecological effect of BT cotton, Monsanto and Mexico issued a 14-page document that showed that condition, that put a condition that whatever the result, I have to share these results, submit these results to the company for their examination, and only after they may approve of this, then I can only send this the documents for publication. Until that time, I cannot even share the data with other scientists. And this is exactly the case in country after country for all transgenic crops, whether it is BT cotton or BT corn or BT soya or Roundup Ready soya or whatever, or Roundup Ready canola. Everywhere it is the same thing. The scientists are imposed with this conditionality that they have to share, they have to submit all their research data to the company for their approval. And obviously, because of this conditionality, some of the scientists who actually agreed to this condition failed to publish any of the results if they were adverse to the company's profit. And this was also published just a few years ago in a special issue of American Scientist magazine. And this American Scientist cover story was, do corporations rule science? And about a dozen scientists wrote about these conditionalities imposed by these corporate sector. So if science is governed and dictated by the corporations, and if the results of this research is also controlled and suppressed by the corporations for their own profit, it's very, very commonsensical to say that there will be no research at all that would prove these adverse effects. And therefore, it's very beneficial for these companies like Monsanto or Syngenta to keep on saying that there is no relevant data to prove that there is no adverse effect. Of course, there cannot be, because scientists have no access to this kind of data, because even if the scientists who have proven this kind of data never see the light of the day, or some scientists who have already published, like Professor Srihari, are ridiculed, and finally this publication, after all the peer reviewed by four stalwart scientists of the same field and experts, they are published, and after that it's revoked. And perhaps the politics is so deeply entrenched and so perverted that the editor of this journal was changed, and this new editor of this journal, immediately after his taking office, he revoked this paper. So it seems that this new editor was appointed specifically to revoke this paper of Srihari, because it's been really a bone of contention and very difficult debacle for the company's profit. The same thing happened with the publication of Ignacio Chapela, who published in Nature who proved that the pollen from GM corn had cross-pollinated, and it affected the non-GM corns 400 to 500 kilometers away in Oaxaca district of province of Mexico. And this was published in Nature after very rigorous peer review by four scientists of the discipline. And soon after that, there was a barrage of opposition and objections from about 60 scientists from all over the world, saying that this was methodologically wrong, the data were wrong, the analysis was wrong, and so on and so forth. And then again, this paper was revoked. The paper was published in 2001 by Twist and Chapela. Chapela was at that time, a postdoctoral fellow and in University of California at Berkeley when I was working there too. And soon after that, this barrage of oppositions of apparently scientists, this paper was also revoked in 2002. And then, if you see this very nice film entitled The World According to Monsanto, it appears that 80% of the emails that were sent to Nature's editor were all generated from one single IP address. And that IP address happens to be the IPR office of Monsanto company. So the same company, using their office computers, generated those, maybe dozens and dozens of false, novel email IDs of ghost scientists, and all of them issued these same kind of letters. And that was really a political pressure on the editor of Nature, who were coerced to finally revoke the paper. And nobody asked the questions of the existence of these scientists who actually wrote these letters. And this is what is science, which is 1,000 light years away from the science that Darwin and Newton had to pursue. So unfortunately, people today still believe that science is objective, science is impersonal and esoteric, and it doesn't affect and is not affected by this kind of politics or commerce. But today's science is just that. Corporate science now dictates which type of research should be conducted, which type of data should be published. And the same comparison and similar effects were with DDT or pesticides or even tobacco. Perhaps you know that tobacco companies have published dozens of ghost-written papers which proved that there is no link between tobacco and cancer.
Yeah.
And some of those papers were even published in prestigious journals like New England Journal of Medicine. And now everybody knows that why those were written and those are published over 4-5 years continuously. Today we accept, after 100 years of research of these effects, now we all have accepted that yes, tobacco is or nicotine is harmful for and affects cancer. But even 20 years back, there were a group of scientists who argued no, there is no evidence, and exactly the same argument that the GM crops are saying.
Okay.
And that's why this precautionary principle must be applied to GM crops, with the assumption that there may be some effect, and until we prove it safe, after a long-term effect, and this long-term needs to be very genuine long-term, because it's not that someone drinks a poison and immediately gets killed off, or it's not that you consume a GM product and the next day the consumer dies off. It's not that. It's just like DDT, for example. The effect of DDT took more than 50 years to emerge, and now we get all the evidence that DDT has, what kind of enormous effects DDT has on biosphere and our health. But it took 50 years to know. So similarly, we cannot predict safety of any GM crop with just 1 or 2 years data and with just one type of model and a very small number of population of these kind of models in one laboratory. It requires rigorous tests on a long-term basis on the presumption that there may be some effect, hidden effect. And until we have applied all the kind of rigors in statistics, we cannot say that it's safe to consume. That's the precautionary principle. Yeah.
Excellent, sir. I know you are short on time. I also wanted to talk about the impact on the farmers, especially financially. But
Thank you. In fact, I was, because of this brevity of time, I'd be happy to have given you some of the concrete references with years and the authors, the full names and the page numbers of these ones for the benefit of the viewers and listeners. But I hope all of the references that I have given you, whether it's Chapela or Priest and others, can easily be found on the net, on the website, and can be downloaded. And also the debate and the special issue of scientific American scientists and others can also be downloaded for free by any reader who has interest in cross-checking my sentences.
Excellent, sir. Sir, hopefully we can have another session where we can talk more on the farmer side and how they are impacted, and also how you do the preservation of these seeds.
Sure.
Okay.
I'll be happy to. Thank you.
Dr. Debal, thank you very much for the time and explaining and going through all these issues.
Okay. Most welcome.
Okay.
Thank you very
much.
Sir.
Friends, if you liked today's show, please go to youtube.com/NRI Samay and click on the subscribe button. You will get notified every time we publish a new show. And you can also like us on our social media sites like Facebook, Twitter, and Google+.